Okay, now let's talk about the dopaminergic pathway that's associated with our reward system. Now, if you haven't already, you should see, if this picture here looks unfamiliar, you should go back to the Dopamine 02 Pathways video and watch that first. But this is where we left off, and I wanted to point out something specific about the dopamine receptors in these two different pathways. And so what are these two different pathways? Well, as we described before, this one from the ventral tegmental area going into the cortex, this is the mesocortical pathway. And the mesocortical pathway is dominated by really exclusively D1 dopamine receptors. And this pathway here, the second one of interest, is the mesolimbic pathway. And it synapses onto neurons with D1 and D2 type receptors. Right, and yeah, let's be clear about that. The neurons in the ventral tegmental area, and the cell bodies are in this area in the midbrain. And their long processes, their axons extend, in the case of the mesocortical, into the cortex, right? So we're going out anteriorly here, and it's sort of, when I curve up here and I come back, this is going more superior. So I'm going to attempt to give a little 3D effect here by making it look bigger, as if it's coming off of the screen out towards you. Right, and in fact, this extends all the way back to the entire cortex here. I don't know if this is doing, if this is helpful or just more confusing, but this, I'm making it thicker here. This is the axon going into the cortex that is above the picture that I drew, into the cortex, right? And there's lots of processes, axons that come off, right? So this is a bundle of many neurons that are coming from the ventral tegmental area. So the medial forebrain bundle, as we call it when it's still in the hypothalamus, and extends out into the cortex. And so all of the neurons that this synapses onto, right, because this is releasing, these are releasing dopamine, and they are synapsing onto target neurons in the cortex, and they are going to have D1 receptors, right? So the receptors are found on the targets, the target neurons, and it's the same for the mesolimbic pathway, that specifically the neurons that it is synapsing on inside the nucleus accumbens in our striatum, and this is sort of just a blown up picture of a postsynaptic dendritic spine, for example, are going to have D1 and D2. But remember, neurons from the VTA are making neuronal synapses. They're also creating those axo extracellular synapses where they're not synapsing on anything. They're just flooding the extracellular space with dopamine. And then also the varicosities, those bulges in the axons, as well, dispensing dopamine. Okay, so now that we have that out of the way, let's talk about conceptually here the reward pathway or system, reward system of the brain, right? And this is composed of the mesolimbic and mesocortical pathways. The mesolimbic and mesocortical dopamine pathways. I just want to make that clear. I know I've said that a bunch of times and it's maybe obvious, but I want to reiterate that this reward system in our brain is a combination of these two dopaminergic pathways. And this reward system is our current understanding of it. We would describe it as a teaching signal. And let me explain what that is. It's as opposed to what it used to be when we were first studying it. It was known as the dopaminergic signals from the VTA to these areas were thought to be a hedonic signal initially. Hedonic, just a word that means having to do with the feelings of pleasure. Feelings of pleasure. That is what hedonic means. So let me maybe sort of illustrate in a very simple way some of the first basic experiments. And these, of course, were done in rodent models. So I'm going to draw a very crude and perhaps offensive for all of those rat lovers out there. Right. So this is supposedly a rat. Okay. So here's our adorable little rat. One of the first subjects here, as we're investigating the effects, the mechanisms, the purpose of dopamine. And so we can put this rat into a cage. And first, let me sort of set up our graph here. So this is time, and I'll draw it over here. This is our dopamine concentrations, and I'm going to draw multiple graphs here. So this is high, and that's low. And so we can, let's say we put this rat into a special cage. And we give it right here, we give it a food reward. Okay. And so we put this rat into a cage, nothing special, and then when it gets the food reward, it gets a surge of dopamine. All right, and so these are gonna be little mini graphs all down here. So this is low, and right, this is high. So we have a high concentration, a little blip here of dopamine, or a significant increase of concentration, not just a blip. And so it was thought like, well, our food reward results in this dopamine surge. Therefore, it must be a hedonic signal, right? It must be dopamine equals pleasure. That this is mediating, this is causing our subjective experiences of pleasure. But they repeated experiments with the rats, and what they found was after they were first introduced to this food reward, this new scenario, and they brought them in the next day, the next trial. And what they see was as soon as they put them into this special cage, that is when they received the dopamine surge. So the special cage. And so how could this be associated with the pleasure of the food reward when dopamine had already peaked and come down to basal levels before the reward was actually obtained? And so this signified that perhaps that this is not the right sort of conceptual way to think about dopamine, that it's not a pleasure signal, that it's not a hedonic signal. And that brought us to our current understanding as dopamine as a teaching signal, as we'll see. I'll sort of walk through it in a conceptual flow chart of how this is. And I want to make one more point here that'll be more salient once we go through the conceptual model of how it's a teaching signal. But essentially, this dopamine surge that's happening right here, what it's doing is it's remodeling our brain to increase the probability of us obtaining the food reward. In the specific of the rat obtaining the food reward. A very interesting thing here. What happens if I also want to point out here that repeated over and over again, what happens eventually is, I'll put a little bit more of an intermediary here, is that the dopamine signal eventually disappears. So it's not constant. They put the rat into the special cage. The rat goes through all the same motions, gets the food reward, but there is no dopamine signal. There's no acute surge of dopamine here. Now, what if we were to, in this experiment, or a different, we'll do a different set of experiments here. What if we make this food reward, instead of the food reward every time, we make it 50-50. Sometimes there's no reward, half the time. The other half the time there is a reward whenever the rat is put into the special cage. And so 100% of the time we get the same dopamine signal. Okay? What happens if we change it to 50% of the time? Any guesses? This is what we see. A massive, right? I'm even bleeding into this other study here. A significant, often doubling increase of dopamine. And this is more persistent over time, where it doesn't disappear as fast. And so what we've done here is, and I'm going to quote Robert Sapolsky, here, a famous neuroendocrinologist, is that we've introduced the word maybe, which has a very powerful effect on our dopaminergic system, as you can see here. And this will make sense in the context of dopamine as a teaching signal. When the food reward is only 50-50, the brain is sending the signal to the dopaminergic pathways saying, this is really good. We need to maximize our probability of getting it again, because this is a pro-survival sustenance, a pro-survival item. It would be great if we could come across again. And so let's maximize our ability to do that. And what dopamine does is it increases the desire, the craving, as well as the memory of the behaviors that are associated with of finding that reward. And so when it's only 50-50, your brain sends a more intense signal because you haven't figured it out yet. There's still more work to be done to increase the probability of finding the food reward more frequently, as close to 100% of the time as you can. And just this little anecdote here of the increased dopamine when you've introduced the word maybe, you might be able to start thinking about how people can become addicted to games of chance, because visiting the casino in Las Vegas and playing the slot machines is, in essence, this introduction of the word maybe in this experiment of giving a rat a 50% chance of getting a food reward, pulling the lever, and anticipating that reward, and not always getting it. And there's a whole industry built around the psychology of people designing gambling games to make people think that their probability of winning is much, much greater than what it actually is. It's actually, one in millions. They do a very good job of making you think that you're almost there, that the probability of you getting a reward on the next pull of the slot machine is much, much greater than just 1 in a million. But let's go on and think about here in a much more boring sort of conceptual map of the reward pathway. Okay, so let's go over a conceptual map. I'm going to write a reward system up here to remind us, and that this reward system is thought to be a learning signal. That is what dopamine's doing in this reward system, rather than being a hedonic signal. It's not responsible for the pleasure itself, but it's responsible for teaching us how to obtain this stimulus again. And so that's what it starts with. It starts with stimulus. In this example, this could be anything, right? This could be chocolate. This could be anything that might. I don't know why I can't think of anything else besides chocolate, but often you think of things that increase organisms' fitness, right? So food, or often, or mating opportunities. All right, but trying to give an example here that will be salient for us as Homo sapiens and modern humans, more accurately to say. And so let me offer this. Let's do this. Stimulus is I'm going to say, well, I'll attempt this. A new crush. Okay, so let's say you've come across a new person that you've never met before, someone that you find very attractive, and someone that you had a very pro-social interaction with. Okay, so there was lots of flirtation going on, and this is someone that you find very attractive, and it seems to be reciprocal. And so this is, there are two things about this new stimulus, about this new crush, is that it's unexpected, and it's what I'm going to call fitness, which is any kind of stimulus that is either going to enhance an organism's ability to survive or reproduce, right? So in this case, it's obviously a signal that's pro-fitness from the perspective of pro-reproductive success. Now, this isn't obviously what you're thinking about mixing your genes with their genes when you're engaging in this flirtatious new encounter. But we've evolved to be attracted to this stimulus in this way because over evolutionary time, it's been selected for because it is more likely to lead to reproductive opportunities. All right. And this is processed and determined by, right, the brain. So the brain processes this information. And ultimately, right, it's your brain that's the one that's determining that this is unexpected, or I guess I should say realizing that this is unexpected, and realizes that this is pro-fitness. And I'm drawing a box around here because this process is very much a black box in terms of how does the brain do this. Well, there's lots of what we don't understand of how it does this, how it makes this determination of this unexpected pro-fitness stimulus. And this is that whether, and I'm not talking about, an attractive person, someone that you think is attractive, but this could be, chocolate. So you've eaten chocolate for the first time, and how does the brain process that, oh, this is good, and we should find more of it? And so your brain determines that this unexpected pro-fitness stimulus is just that, very good for us, so we need to find more. We need to experience this stimulus more, right? And organisms that have evolved this pathway and this drive to seek out this new experience are more likely to be afforded reproductive opportunities, right? So this is the sort of evolutionary explanation of why it feels good to interact with other people that you find attractive. And so this brain, the specific brain processes stimulates the ventral tegmental area. And it's the VTA that then goes on to, and it secretes dopamine, right? Or it transmits and releases dopamine, both in the nuclear accumbens and also to the medial prefrontal cortex, right? Nucleus accumbens being part of our mesolimbic pathway, and the medial prefrontal cortex being part of our, if you remember, part of our mesocortical pathway. Now, it's this signal here, this increase of dopamine. That increase of dopamine to these areas is not what gives us the hedonic signal. I mean, there clearly is a hedonic signal associated with this new stimulus, but this signal is, the hedonic signal is largely thought to be driven by things like endorphins, opioid, interaction with the opioid receptors through endorphins, for example, and no longer thought to be due to dopamine. Right? Dopamine is not the pleasure signal. And so what the dopamine does is it causes remodeling. It causes remodeling. And how does it do that? Well, we had a number of videos about that. It does that through neuromodulation of various kinds. And so this remodeling, what is it responsible for? It's responsible for two things. So the remodeling is responsible for one, increasing our craving and increasing the wanting of the stimulus. So the brain says, this never had this stimulus before, but it is good. And we need to maximize our chances of finding this stimulus again, which means interacting, in this case, interacting with this individual again. And so it increases that. It also facilitates and increases developing the memory, right? Or in specifically here, I should draw this like a subcategory, the associations. The associations, including the cues and the behaviors that around this stimulus, that preceded the stimulus, that led to this stimulus. And so this remodeling not only helps creates a drive to seek the stimulus, but also helps you to remember how to find that stimulus again. So you remember, the where you were, the time of day, and the behaviors, right? Did you tell a certain kind of joke, or did you look a certain way, act a certain way that seemed to be to lead to this interaction, to make this person laugh, or to be flirtatious in a way? So dopamine helps you remember, how can I maximize this, finding myself in this situation again, and interacting with this person again, right? And all of these two things, right, are meant to maximize finding the stimulus again. And I keep saying that, like, finding the stimulus, I don't mean this in the case of, like, I'm not describing the person as a stimulus, but it's the interaction with the person was the stimulus. The sort of the flirtatious first meeting is the stimulus that that the brain is seeking again, and changing, remodeling, so that it's more likely to find this again. And one of the, as we'll see soon, one of the dangerous things about addicted drugs that have a high risk of addiction is that they bypass all of this part right here. Addictive drugs bypass this part right here. And what they do is that the addictive drugs just go straight for the VTA. And what they do is they directly act on the VTA neurons to increase the release of dopamine, or in some cases they act on the neurons that act on the VTA to increase dopamine. So these addictive drugs are not bypass this brain, a higher cortical brain processing of the information to determine if the VTA gets activated. It just activates the VTA. And this is important and perhaps subtle, but thinking about in this situation here, what happens is that over time, once your brain goes through this remodeling, and once you successfully find and are able to get this stimulus over and over and over again, the brain says, okay, we've done our job. We don't need dopamine surging anymore because we figured it out. We don't need to keep remodeling the brain to increase craving. We don't need to keep remodeling the brain to enhance our memory, to maximize finding the stimulus, because we've done it. We figured it out. We know it's good. We know how to get it. That's it. And in the chapter that you read in the Molecule of More text that you wrote, that you read, this is exactly what happened in that sort of interwoven narrative of the couple that first met, and then over time the dopamine sort of wears off. And this is exactly what happens naturally with any new unexpected stimulus that is determined to be pro-fitness. Dopamine increases to maximize your chances of obtaining that stimulus again. And then once you do repeatedly over time successfully, and you know how to do it, that effect wears off. That craving and wanting wears off because you're no longer remodeling for it. It goes back to baseline. Yeah. Okay, so let's take transition now to talking more in depth about addictive drugs and the specific mechanisms of what they're doing along these dopaminergic pathways.